Our third presenter, in fact, there's two presenters, Sharifa Sahai from Harvard Medical School and Ana Ramos-Guerra from the Polytechnic University of Madrid will talk about automated detection of BK virus in H&E whole slide images using weakly supervised deep learning and interpretable morphological biomarkers.
Hello. This work is a collaboration between the Harvard Medical School and the BIT lab at UPM in Madrid. Our work is entitled Automated Detection of BK Virus in H&E Host Slide Images Using Weakly Supervised Deep Learning and Interpretable Morphological Biomarkers. I'm Sharifa Sahai, and this is Ana Ramos. So some clinical context behind BK virus. There is over a 90% prevalence rate of BK virus in the human population. However, it's usually asymptomatic, but reactivates under immunosuppression. This is a huge threat for kidney transplant recipient patients. Over 10% of recipients will get BK virus. And this untreated can lead to irreversible graft damage. Therefore, it's a critical diagnostic challenge, especially to determine BK virus from rejection, which require opposite treatments. BKV requires lowering immunosuppression, while rejection requires increasing immunosuppression. So some background on how BKV is usually diagnosed is first you would have to do a kidney transplant biopsy. From this biopsy, a set of routine stains, including H&E, PS, trichrome, and Jones silver are produced. In addition, serological assays such as a BKV titer are done. If suspicious in the titer, then an additional IHC stain, the SV40 stain, is required. And this stain, under visual examination by a trained pathologist, will lead to the diagnosis of BKV or not. However, this approach is quite time-consuming, as the IHC stain is an additional stain and is not routine. It has a higher cost. In addition, an additional biopsy might be required if there's limited tissue available, and it requires specialized equipment, which may not be available in all centers. In contrast, our method BK Vision utilizes the most commonly utilized stain, which is H&E. And from that, we apply an attention-based multiple instance learning network to also extract human interpretable features in addition to doing classification. This method is fast, automated, and highly interpretable due to the use of attention maps to create the markers, and it is low cost because you can use the routine stain. Some background on the data utilized for the study. We have over 1,000 patients that were utilized, over 4,000 whole slide images, around 200 IHC. And we also note that for the BK positive slides, there were only about 4% were BK positive. And we employ several computational methods to address this. So our model is a two-step pipeline. In the first stage, the H&E images undergo our ABI-ML model, which is based on the CLAM architecture from the Mahmood lab, where which is my lab. From that, we can classify if the images are BKV positive or not, and we can also, from those positive images and negative images, collect a set of high attention patches which correlate with the viral stain, and negative patches. In the second stage of the pipeline, we take these patches and we register them with the corresponding IHC, and then we can do quantifiable analysis using and create human interpretable features such as area and size of the cell, texture analysis, and see which ones are differing the most between the two populations. We utilize several widely available open source models for this, including VALIS for whole slide image registration, HooverNet for nuclei segmentation, and histocartography for generating the human interpretable features.
To diagnose BKV infection, we compare different patch encoders, ResNet50 that was pre-trained on ImageNet, and 3 computational pathology foundational models, which are cTranspath, Uni2, and Conch. Since our dataset was highly imbalanced, we also tried different sampling rates to overcome this, and we observed that the Conch patch encoder combined with the extreme upsampling obtained the best results for across most of the metrics, as we can see here. And we believe this is because Conch was trained on more diverse data and because of the low prevalence of the BKV positive cases. To identify morphological features predictive of BKV infection, we visualized the attention scores given by the model. As we can see in the image, we have an example of an HNA stain, its attention map, and an IHC closer to the HNA. In the red boxes, we can see an example of a high attention patch, which includes positive nuclei, as we can see in the IHC slide, as brown stem cells. In the blue boxes, we can see an example of a low attention patch that includes no infected cells. These attention heat maps are also quite informative because the pathologists could visualize it and see that in the high attention regions, there were some signs of infection, for example, tubular cell injury. At the nuclear level, we extracted the pathomics features from the high attention patches of positive cases, and we compare them to the pathomics of cells from negative cases. And we observed that the features that were mostly significantly related to the infection were mostly related to the cell texture, mainly the chromatin distribution changes that were caused by the virus. This is known as the ground glass phenotype, where the cells appear more homogeneous and diffuse, as we can see in the examples on the right, where we compare the positive patches, high attention patches, and the negative patches in blue. Other features that were significantly related to the infection were mainly related to the cell size and shape. Specifically, the infected nuclei were mostly larger than the non-infected nuclei. To validate this, we asked an expert pathologist to manually segment the infected nuclei here in yellow with the help of the closest IHC and compare the area with the non-infected nuclei. As we can see in the histogram, the infected nuclei are clearly larger than the non-infected ones. To sum up, we accurately predict BKV infection only using routine HRA, reducing the reliance on SB40 IHC, and we also identified morphological features such as the cell area and the chromatin density, validated also by experts. We want to formally evaluate the identified biomarkers across diverse populations, also to validate them in a real-world setting. We hope BK Vision serves as a blueprint for interpretable computational pathology tools that can be applied to other diagnostic challenges beyond BKV detection. We would like to thank our respective labs in Harvard and UPM, and also our mentors and collaborators, and especially the pathologists that help us to validate our findings. Thank you all.